Let's proceed with gametogenesis, spermatogenesis, and oogenesis. Starting with oogenesis, the formation of an oocyte. So what happens is in the second week of embryonic development, the epiblast has been formed, and the primordial germ cells are going to come from the epiblast and then migrate in from the by the fourth week to the yolk sac, where the developing gonads are going to be. And then in the sixth week, they are going to reach the ovaries, of course, and differentiate into oogonia. The primordial germ cells and oogonia, they've got 46 chromosomes and a diploid number of DNA. When they reach, of course, the ovary, they'll start to undergo meiosis 1, and these are the oogonia because the primordial germ cells are differentiated immediately they get to reach the ovary. They start undergoing meiosis 1. And in meiosis 1, there's going to be DNA replication happening, and the primary oocyte is going to be formed. Now, because you have replicated this DNA, you started with this number, it has made a copy of itself. So it is now twice. The chromosomes, they are still 46, but the DNA is now 4N. The same information, but twice. So that's why we can see from 2N to 4N, but the 46 is not changing. So those are the primary oocytes. The primary oocytes, of course, all of them are going to be formed by the fifth month of fetal life, just inside the womb as the girl child is developing. So by birth, there's going to be no oogonia present because all the oogonia would have been converted into primary oocytes. And then the primary oocytes is going to remain dormant in prophase 1, specifically the diplotin stage, from about month 5 until puberty. So meaning from month 5 till puberty, you only have primary oocytes in a girl child. There is no ovum. There is only a primary oocyte. So after puberty, then 15 to 20 primary oocytes are going to be ovulated each month. Each month 15 to 20. Each month 15 to 20. Like that. So of these 15 to 20 primary oocytes ovulated every month, only one is going to reach full maturity. And that's the one actually which is going to be ovulated. 15 to 20 are just going to be released to undergo development, but only one is going to mature. During the ovarian cycle, of course, luteinizing hormone is the one which is going to be responsible in getting to recruit all these primary oocytes to be able to complete meiosis 1. Remember, they were arrested by month 5. And up to puberty, this is when they've been released by luteinizing hormone to be able to complete meiosis 1 from the diplotin stage where they were arrested. And so when they get to grow, they are going to form two daughter cells, the secondary oocyte and the first polar body. The secondary oocyte has got 23 chromosomes and 2N DNA. We can see the DNA has reduced from being 4N to 2N, and also the chromosome number has reduced from being diploid to haploid. And the first polar bond we should consider is being formed. Now, when the daughter cell, which is the primary oocyte, starts to undergo meiosis, it's going to form two cells, secondary oocyte and polar body. Because the polar body does receive little nutrition support, it is going to degenerate, and the secondary oocyte is the one which is going to continue. So the next that happens that the secondary oocyte is going to now begin meiosis 2. When the primary oocyte has been released during the period of menstruation, that period, or let me just say during puberty, what is now going to happen is that the secondary oocyte is now going to continue with meiosis 2. But it will not also finish meiosis 2. It is going to be arrested in metaphase, that is just 3 hours before ovulation. Secondary oocyte is going to remain arrested until fertilization happens. So if fertilization does not happen, then this secondary oocyte is going to be released in menstruation. So during menstruation, what is released is actually the secondary oocyte and not anything else. So if this secondary oocyte had to mature, it's going to form a mature oocyte at fertilization, and that is the one which is going to complete meiosis 2. Secondary mature oocyte has got 23 chromosomes, and then it has reduced also to 1. And remember, secondary oocyte was 23 and 2, and this one is 23 and 1N. And then two more polar bodies are going to be formed from that first polar body which was formed from the primary oocyte. That polar body will also form two more polar bodies. And so we're going to have a total of three polar bodies. So of all these polar bodies, why are we having all these? Because in a female, you have formed X, 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 X. Three of these are going to be inactivated. Only one is going to be active. That's the one which gets to mature. So now, the primary oocytes which are going to be formed by month 5 are around 7 million, and at birth, only 2 million are going to be present. The other 5 million is going to undergo atresia. They're going to be destroyed. But during at puberty, not all the 200 million will reach puberty. I mean, not all the 2 million will reach puberty. Only 40,000 are going to be present at puberty, because the rest are also going to be degenerated by atresia like that. The secondary oocytes, of course, if each year 12 are going to be ovulated in a normal person. This might not always be true, because if conditions such as metrorrhagia, menorrhagia, oligomenorrhagia, dysmenorrhagia, so these are able to affect the secondary oocytes which are being released. So that might affect the number. But for a normal girl, we expect that 12 secondary oocytes are going to be released each year. So this means that up to 480 are going to be released the entire reproductive life of the woman. Think of it, if a reproductive life of a woman is 40 years, say this lady started menstruating at, let's say, 12 years, and then the menopause, say, happens at somewhere 50 something. So if you calculate the years between here, that would be 40 years approximately, and then you multiply by 12, only 480 are going to have been released. Okay. So this number, of course, is not like really the only number possible, because others start late menstruating and then menopause reaches fast. Others will start menstruating earlier and then menopause also will be late. So depending with how many years are going to be present, this is more like just an average number. So we're able to calculate, say, if someone started menstruating at 16 and then menopause happens at, let's say, 45, how many would have been released? So just multiply the years between and then times 12 if it was a normal menstrual cycle. So this is what is happening. You have got first the primordial germ cells converted into oogonia in the ovary, which has got 2N. Oogonia after meiosis will go into primary oocyte, which has got 4N. And then the primary oocytes will go into secondary oocyte, which has got 2N. And then secondary oocyte, it will go now into a mature oocyte, which has got 1N. Mature oocyte, if it is fertilized, you're going to have 2N, because 1N is coming from the sperm and then 1N is coming from this ovum. Let's talk about spermatogenesis. So the one we are calling as the mature oocyte is what we call an ootid. So let's talk about spermatogenesis. Just like also oogenesis, you have got first spermatocytogenesis. This is the formation of spermatocyte. And then we're going to have meiosis and then spermiogenesis. So this is a division of spermatogenesis. So spermatocytogenesis, you have got the primordial germ cells 46 and then 2N, which are going to arrived in the yolk sac and in the test by week 6, in the yolk sac by week 4, and then they are going to migrate to the test by week 6. Where are they coming from? They are coming from the epiblast, where they were formed in the second week. So this is going to, when they arrived in the test, they are going to remain dormant. These are the primordial germ cells. They are the ones which remain dormant until puberty. Different from a girl, it is a primary oocyte which remain dormant till puberty. For a boy, it is the primordial germ cells. And then at puberty, primordial germ cells are going to differentiate into type A spermatogonia, which has got 46 and 2N also. So type A spermatogonia, these are stem cells which are going to carry these cells, because the process of spermatocytogenesis happens for the rest of the lifetime of a man. It's not like in a girl where if it finishes, then it has finished. So the type A spermatogonia is now going to differentiate into type B spermatogonia. It is also having 46 chromosomes and then 2N. Meiosis is going to happen. Now, what happens in meiosis is that type B spermatogonia is going to undergo replication, and they are going to form primary spermatocytes 46 and 4N because of replication 4N. And then the primary spermatocytes are going to complete meiosis 1 to form the secondary spermatocytes 23 and 2N. The secondary spermatocytes are going to complete meiosis 2 to form spermatids, just like the ootids, that is 23 and 1N. So when we form the spermatids, these are not that mature. Even if they are already sperms, they need to undergo some morphological changes. The changes in their structure and in their shape. That is now called spermiogenesis. So when these spermatids undergo post-meiotic series of morphological changes, then they're going to become fully mature. So there's going to be formation of the acrosome, condensation of the nucleus, and formation of the head, neck, and tail. So this period of spermiogenesis lasts about 64 days. Now, they don't give space for each one to complete a cycle. One spermatid can be on day 6, while another one is on day 27, another one is on day 30. So they are happening continuously, and each one, the period continues to go like that. So the fact that people are reaching grade 12 doesn't mean they are those who are not, there are no people in grade 1 or grade 2. So it is all happening simultaneously. So this is what happens: acrosome formation, nucleus condensation, of course, also the shedding of the excess cytoplasm. The newly ejaculated sperm are incapable of fertilization. This is what we need to know because they undergo capacitation. When a sperm has been formed, there's going to be a glycosyl transferase and a plasma protein coating on the head. So this is to prevent these sperms from searching for the ovum in the male reproductive tract. So if these sperms are supposed to fertilize the egg, they are supposed to undergo capacitation. So when they have got the glycosyl transferase and the plasma protein coating, that is called decapacitation. You are removing their capacity to fertilize. And then they need to undergo capacitation for them to have capacity to fertilize. And this happens in the female reproductive tract after they've been released. So this is a sperm formed. It has got the head, the neck, the middle piece, and the tail. The tail has got a plasma membrane. The middle piece has got a lot of mitochondria. And then the head contains the nucleus. And then at the end, the front part of the head, we have got the acrosome. This is the one responsible for digesting the zona pellucida of the ovary for the sperm to be able to enter and fuse with the ovum. So here is a spermatid formed. You can see it is not mature. This can't fertilize. So it's to start to undergo morphological changes. As we can see, we have got the centrioles, which are simply microtubules forming the flagellum or the tail. So the tail is formed from microtubules in case a question came. And then you can see the cytoplasm is also going to the tail, okay, like that. So this excess cytoplasm is going to be shed off, and then you are going to have now a fully developed spermatocyte. So let's consider some questions. Which of the following is a major characteristic of meiosis 1? Splitting of the centromere, pairing of homologous chromosomes, reducing the amount of DNA to 1N, achieving the diploid number of chromosomes, producing primordial germ cells. Which one would we go for? So we know when we're talking about meiosis 1, splitting the centromere happens during meiosis 2. Meiosis 1, you simply split homologous chromosomes. Pairing of homologous chromosomes, of course, this is true. Reducing the amount of DNA to 1N, we saw this wasn't happening during meiosis 1, but of course, it happens by the end of meiosis 1 during telophase 1. You are going to have a diploid number. But this 1N, remember, it was the formation of a spermatid, and also that was happening after second meiosis. Achieving diploid number, you don't achieve the diploid number by meiosis because meiosis is a reduction. Producing primordial germ cells also doesn't happen. So the answer here is B, pairing of homologous chromosomes, specifically during prophase 1. A normal somatic cell contains a total of 46 chromosomes. What is the normal complement of chromosome found in a sperm? So we know a sperm, this is only having 23 chromosomes. Or the 23, 22 are autosomes, and then only one is going to be a sex chromosome. So it is 22 autosome plus a sex chromosome. That is A. 3. Which of the following describes the number of chromosomes and amount of DNA in a gamete? Now, when I'm talking about a gamete, what are we referring to? Is it a primordial germ cell? Is it oogonia or spermatogonia? Is it primary oocyte, secondary oocyte, primary spermatocyte, secondary spermatocyte? Are we talking about a spermatid otid? What are we talking about? So a gamete is what is produced at the end of the process. That is either an otid or spermatid. And we saw it had 23 and 1 N. 23 chromosomes and 1 N. So the answer is C. Which of the following chromosome composition in a sperm normally results in the production of genetic female if fertilization occurs? Now we've already said that these gametes have got 23 chromosomes, 22 which are autosomes, and then one which is a sex chromosome. Now, if we want to form a female, we need to have X, because if we have got Y in that sperm, then definitely it is a male that is going to form. So it is 22 autosome plus an X chromosome. Therefore, the answer here is E, 22 autosomes and an X chromosome. So this one, 23 is not correct. 22 homologous not correct. So that is it. In the process of meiosis, DNA replication of each chromosome occurs, forming a structure consisting of two sister chromatids attached to a single centromere. What is this structure? So sister chromatids attached to a single centromere, we call that as a chromosome. But what chromosome? Is it a duplicated chromosome, two chromosomes, or a homologous pair? Now, these, as we've been taught from the question, DNA replication. So we have simply replicated the same thing. So it is actually the same thing. It is just a duplicated chromosome. Homologous chromosomes, one come from the mother, one come from the father. It doesn't happen in replication. All right. So the answer here is A. All primary oocytes are formed by. So we already mentioned all primary oocytes are going to be formed by month 5 of fetal life, not of infancy. Infancy is when a child is already born, but fetal life, this happens in the uterus. When does formation of primary spermatocytes begin? So for primary spermatocytes, we said the primordial germ cells are arrested not until puberty reaches, and then the primary spermatocytes are going to be formed. So they are formed at puberty. All right. Question 8. In the production of female gametes, which of the following cells can remain dormant for 12 to 40 years? 12 to 40 years in the production of a female, important word there. So what happens in the female? What is going to be produced is going to remain dormant for 12 to 40 years. So we already saw when the primordial germ cells have arrived in the ovary, they are going to start converting into the primary oocytes. The primary oocytes by month 5, they are going to remain dormant, not until someone reaches puberty. And they'll just be released 15 to 20 each month, 15 to 20. So what happens is that there are some which would have been formed by month 5, but even up to puberty they've not been released. So they wait even up to maybe the time when someone is 45 years, that's when they're going to be released. So the primary oocytes are going to be able to stay from 5 months intrauterine even up to 45, 50 years without being produced. They are going to remain dormant. So primary oocytes. In the production of male gametes, which of the following cells remain dormant for 12 years? Primary germ cell, primordial germ cell, primary spermatocyte, secondary spermatocyte, spermatid, sperm. Which one is going to be correct? So in a male, we said that the primordial germ cells are going to be dormant not until someone reaches puberty, and puberty in a male around 12 years. So here it is a primordial germ cells, different from a female. Approximately how many sperms will be ejaculated by a normal fertile male during sexual intercourse? So a normal ejaculation is around 2 to 6 mils, and 1 mil of each ejaculation contains about 50 to 100 million sperms. So say a person ejaculates 2 mils times 100 million sperms, that would be 200 million sperms. If they ejaculate 6 mils, that would be 600 million sperms. So it is about between that number. So what we can simply pick is the highest that we have here, about 350 million sperms being ejaculated. because below 10 million, then this man is impotent. They can't have a child. A young woman enters puberty with approximately 40,000 primary oocytes in her ovary. About how many of these primary oocytes will be ovulated over the entire reproductive life of the woman? We already said that by the time a girl is reaching puberty, she had 40,000. Now, about how many of these primary oocytes will be ovulated the entire reproductive life of the woman? So if we are able to do a calculation, we said, of course, we first have the primary oocyte each month, about 15 to 20 are being released. So let's say 15 have been released, and the following month also 15 have been released. The other month also 15 have been released. So let's multiply that number. 15 multiplied by, let's say in a year I've got 12 months, by 12. That is 180. Let's say the reproductive life of this girl is 40 years. So if you multiply by 40, that is going to give about 7,200. 7,200, around that number. Now, we said of these which are going to be released, how many are going to be ovulated? Only one is ovulated each month. So this number that you've gotten, 7,200 divided by 12. Okay, if we divide it by 12, that will give us around 600. We already mentioned the number 480, but just for calculation's sake, in case the multiple choice came and other values were showing up. So in each month, only in each month, only one is ovulated, and there are 12 months. So simply say 1 times 12. So 12 have been released in each year. So let's say the reproductive life of this girl is 40 years. 12 times 40, that is the one which gives us 480. But the reproductive life, some people might start menses late and then end late. So it might be more or less, but in these ranges of 480 being ovulated. So primary oocytes produced and those which are ovulated, these are two different questions if they're asked. Primary oocyte release are 15 to 20 each month. So you multiply that number by 12 to calculate how many have been released in a year, and then multiply by 40 to find how many have been released in the entire lifetime. But for the secondary oocytes, or those which have been ovulated, is simply 12 times 1 and then times the reproductive age. Female sex can be diagnosed by noting the presence or absence of the barb body in cells obtained from the amniotic fluid. What is the etiology of the barb body? So a barb body is simply the inactivation of an X chromosome. You know that in a male, you are going to form X and Y. So since you only have one X and one Y, you don't have a barb body in a male. But in a female, you are going to have one X, 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 four of them. So what happens is that three of them are going to be inactivated because only one is going to be able to work. So there are three bar bodies. So if someone has got XX, meaning that one bar body, one X is going to be a bar body, and then one X is going to be the one working. So for a female, they have got one X bar body. For a male, there is no bar body. So the answer is inactivation of one X chromosome. That is a D. Question 13. How much DNA does a primary spermatocyte contain? How much DNA? Primary spermatocyte. So already mentioned, primary spermatocyte is 4N. Primordial germ cells and spermatogonia, it is 2N. Primary spermatocyte 4N, secondary spermatocyte 2N, and then mature spermatocyte or spermatid, that is 1N. During meiosis, pairing of homologous chromosomes occur which permits large segments of DNA to be exchanged. What is this process called? The pairing of homologous chromosomes to have the exchange of DNA. Is this synapsis, non-disjunction, alignment, crossing over, or disjunction? So this is a synapsis. The pairing is a synapsis. Crossing over is when these two have crossed, like that is crossing over, they've crossed somewhere. Now the actual pairing together is synapsis. The coming together, the pairing together is synapsis. During ovulation, the secondary oocyte resides at what specific stage of meiosis? During ovulation, the secondary oocyte resides at what specific stage of meiosis? So we mentioned this is at metaphase of meiosis 2. So there are two arrests in a female at prophase 1 in diprotein and metaphase 2, but in a male there is only one arrest that happens in prophase until puberty. Concerning maturation of the female gamete, which is oogenesis, when do the oogonia enter meiosis 1 and undergo DNA replication to form primary oocytes? When do the oogonia enter meiosis 1 and undergo replication to form the primary oocytes? So the primary oocytes are going to be formed during fetal life. By month 5, all of them would have been formed, and then they're going to be arrested until puberty. Where do the primordial germ cells initially develop? In the gonads at week 4 of embryonic development, in the yolk sac at week 4 of embryonic development, in the gonads at month 5 of embryonic development, in the yolk sac at month 5 of embryonic development, in the gonads at puberty. What do you think? Where are the primordial germ cells going to initially develop? So primordial germ cells come from the epiblast formed in the second week. They start migrating to the yolk sac by the fourth week, and then they are going to start also going to the gonads by week 6. So yolk sac week 4, and then gonads, that is in the ovary or test, by week 6. So which one is going to be the answer? The answer is going to be B, in the yolk sac at week 4 of embryonic development. And so this is it for this tutorial. Thank you so much for being present.